Okay, let's unpack this. Every day we eat, right, and our bodies just perform this incredible feat of engineering to process that food.
It's pretty amazing.
But how often do we really stop and think about the complex journey it takes, once it leaves our fork?
Not often enough, probably. We just expect it to work.
Exactly. So today on the deep dive, we're taking a really close look at Chapter 37 of Understanding Pathophysiology. We're going to guide you step by step through the intricate structure and surprising functions of your digestive system.
Yeah, it's more than just plumbing.
Right. Our mission here is to explore everything, from the first bite all the way to its vital immune role. We want to give you clear, step-by-step insights without getting, bogged down in jargon.
And what's truly fascinating here is just how much coordinated effort goes into something we mostly take for granted. This system isn't just about absorbing nutrients. No, it's a master regulator for immunity for energy balance, with incredible precision and coordination at every single step. It's really quite something.
And that coordination, that's exactly what makes it so fascinating, especially when you think about just how many processes are happening all at once. So let's start with the main highway. The gastrointestinal or GI tract, think of it like one long hollow tube.
Right, mouth to anus.
Where food is ingested, propelled along, broken down mechanically and chemically, nutrients get absorbed, waste is eliminated, and all while protecting us from invaders. That's a lot.
It is a lot. And what's really cool is that this highway isn't just a simple pipe. When you zoom in, it's actually incredibly complex. It's built with 4 concentric layers, almost like a fancy garden hose with specialized linings.
Okay, 4 layers. What are they?
So from the inside out, facing the food, you've got the mucosa.
Yeah.
Beneath that, the submucosa. Then comes the muscle-rich muscularis. It's for movement.
Makes sense.
And finally, the outer protective layer, the serosa. But woven within these layers, there's this incredible local nervous system. It's called the enteric plexus. People often call it the gut brain.
The gut brain, I've heard that.
Yeah. It controls movement and secretions locally, but it also talks to your wider autonomic nervous system. So if you could picture a diagram of the GI tract wall, like in figure 37.2, you'd see these distinct layers stacked up, and then these tiny nerve networks kind of sandwiched between them, like miniature command centers, managing the whole process. Digestive glands also empty right into the central space, the lumen, through little ducts.
That's a great visual. Okay, let's begin our journey right at the starting line. The mouth and the esophagus.
Right.
It all kicks off in your mouth with mastication, that's just chewing, right? With your 32 permanent teeth?
Hopefully 32.
Hopefully. And as you chew, your taste buds are working overtime, detecting salty, sour, bitter, sweet, and that 5th taste.
Umami. Savory.
Umami, right. These tastes and the smell of the food, too, they're like the opening act. They signal the rest of your digestive system, get ready, food's coming.
It primes the pump, essentially.
And it's not just chewing. Salivation is super crucial. We have 3 main pairs of salivary glands: the submandibular, sublingual, and parotid glands.
Yep. Constantly working.
They produce about a liter of saliva every day. If you were to picture them, maybe like in figure 37.3, the parotids are kind of in front of and below your ears.
Yeah, the biggest ones.
The submandibular glands are under your jawbone, and the sublingual glands are right under your tongue.
And that saliva is your mouth's clever multi-tool. It's mostly water, sure, but it contains salivary alpha amylase. That enzyme starts breaking down carbohydrates right there in your mouth.
Instant digestion.
Pretty much. But here's something neat. Saliva has bicarbonate in it. That keeps the pH around 7.4, which neutralizes bacterial acids. So it's literally fighting cavities before you even brush.
Wow, okay.
Plus, it's got mucin for lubrication, making food easier to swallow, and immunoglobulin A, or IgA, that's an antibody, providing local immune defense right in your mouth. It's a microscopic powerhouse. And interestingly, like shown in figure 37.4a, its electrolyte concentrations actually change depending on how fast it's flowing. Faster flow, like when you're eating, means less time for modification, making it more like plasma.
Fascinating. Okay, so after the mouth, the food needs to get down to the stomach. That's the job of the esophagus.
Right, the food chute.
It's a muscular tube about 25 centimeters long. What's unique is its muscle setup. The top third is striated, like skeletal muscle, so somewhat voluntary.
Right, the initial push.
But the lower 2/3 are smooth muscle, totally involuntary. And food doesn't just, fall down. It's actively moved by peristalsis.
Those wave-like contractions.
Exactly. Coordinated muscle contractions and relaxations. That's why you can actually swallow food upside down, apparently. Though maybe don't try it right after dinner.
Probably wise. And swallowing itself happens in two really seamless phases. First, there's the voluntary part, the oropharyngeal phase. Your tongue shapes the food into a ball, a bolus, and pushes it back.
Okay.
Crucially, your epiglottis, that little flap, covers your windpipe. That stops food going down the wrong way, prevents aspiration. All that happens in less than a second.
Super fast.
Then the involuntary esophageal phase kicks in. Waves of relaxation travel ahead of the bolus, opening the way, followed by those peristaltic contractions, pushing the food down towards the stomach, maybe 2 to 6 centimeters per second. There's primary peristalsis, triggered by the swallow, and secondary peristalsis if food gets stuck.
And there are gatekeepers too, right? Sphincters?
Absolutely crucial. The upper esophageal sphincter at the top prevents you from swallowing air. And the lower esophageal sphincter, or LES, sometimes called the cardiac sphincter, is at the bottom.
That's the one that causes heartburn if it doesn't work right.
Exactly. It prevents stomach contents, including acid, from refluxing back up into the esophagus and causing that caustic injury. Its tightness, its tone, is regulated by nerves, like the vagus nerve, and hormones like gastrin, progesterone, and secretin.
Okay, so the food has navigated the esophagus and arrived at the stomach.
The churning cauldron.
Huh. Yeah, picture a J-shaped, hollow, muscular organ tucked just below your diaphragm. Its main jobs: store food temporarily, secrete digestive juices, mix everything up vigorously.
Like a cement mixer.
And then propel that partially digested mixture, now it's called chyme, this thick liquid paste, into the small intestine. Functionally, we can divide it into areas. There's the fundus at the top, the main body, and the antrum down at the bottom near the exit.
And if you can see inside, like in figure 37.5, when it's empty, the lining has these prominent folds called rugo.
Ah, so it can stretch.
Exactly. Allows it to expand dramatically after a big meal. It's built tough, too, with 3 layers of smooth muscle, longitudinal, circular, and an extra oblique layer, which get thicker towards the antrum for really powerful mixing.
Does much get absorbed there?
Surprisingly little, actually. Mostly just things like alcohol and aspirin, which are lipid-soluble and can slip through the lining, but not major nutrients.
And it must need a good blood supply for all that activity.
Oh, definitely. It gets a rich supply, mostly from the celiac artery. And the blood draining out goes into the hepatic portal vein, heading straight to the liver. If you remember that splanchnic circulation diagram, figure 37.6, you see how the stomach is part of this whole network feeding the liver.
Right. And it's wired up, too.
Highly wired. It gets signals from the main nervous system, the vagus nerve and sympathetic nerves from the celiac plexus. But it also has its own intrinsic nerve networks, those enteric and submucosal plexuses, the gut-brain we mentioned.
Yeah.
This gives it a lot of autonomy over its functions.
Okay, here's where it gets really interesting. Gastric motility, all that churning and moving, how does that actually work?
Well, first, as you swallow, the top part of the stomach, the fundus, relaxes to make room. That's receptive relaxation, helped by hormones like gastrin and cholecystokinin.
Making space.
Yep. Then, these powerful peristaltic waves start sweeping from the body down towards the antrum. They happen about 3 times a minute. These waves get stronger as they reach the antrum.
So they're mixing and grinding.
Exactly. And as the wave hits the near-closed exit, the pyloric sphincter, most of the chyme is forced back up into the stomach body. That's called retropulsion. It's fantastic for mixing and breaking down food particles.
Okay, so it mixes, then how does it empty?
Gastric emptying is very carefully controlled. The rate depends on the volume of food in the stomach, its osmotic pressure, and its chemical composition. Things like fats or very concentrated solutions slow down emptying. The stomach basically tells the small intestine, hold on, handle this bit first. Hormones like gastrin and mopilin speed things up, while sympathetic nerves slow it down.
Makes sense. It needs coordination. Now, what about the secretions? The gastric juices?
Ah, yes, the stomach's chemical cocktail. It includes acid, hydrochloric acid enzymes like pepsinogen, protective mucus, other enzymes, hormones, and 2 really vital substances.
Which are?
Intrinsic factor, which you absolutely need to absorb vitamin B12 later on, and gastroferrin, which helps with iron absorption.
So not just digestion, but helping absorb key nutrients too.
Right. And the release of these secretions is cleverly timed in 3 phases, all geared towards getting acid released when needed. First, the cephalic phase. Just thinking about, smelling, or tasting food triggers signals via the vagus nerve.
Before you even eat.
Exactly. Then the gastric phase starts when food actually enters the stomach and stretches it. And finally, the intestinal phase kicks in, when partially digested proteins and histamine reach the small intestine, providing a sort of feedback loop.
Okay. And where do these secretions come from? Specific cells?
Yes. If you look deep into the stomach lining, especially in the fundus and body, you see these things called gastric pits. They lead down into tubular gastric glands. Figure 37.7 shows this nicely. You find different cell types there.
Nice.
Chief cells make pepsinogen, the inactive form of the protein-digesting enzyme pepsin. Parietal cells are the stars making hydrochloric acid, HCl, and intrinsic factor. There are also G cells making the hormone gastrin, and other endocrine cells releasing things like histamine and somatostatin.
Let's focus on the acid from those parietal cells. Why so much acid?
Well, that HCl does several key things. It dissolves food fibers, making them easier to break down. It acts as a potent bactericide, killing off many harmful microbes you might swallow. And crucially, it converts inactive pepsinogen into active pepsin.
So it activates the main protein-digesting enzyme.
Precisely. Pepsin needs that acidic environment to work. The mechanism for acid production itself, shown in figure 37.8, involves pumping hydrogen ions into the stomach lumen in exchange for potassium, and chloride ions follow. Interestingly, when the stomach is secreting a lot of acid, the blood leaving the stomach becomes slightly more alkaline, the alkaline tide.
What stimulates this acid secretion?
Several things. The vagus nerve, acetylcholine, the hormone gastrin, and histamine are the main drivers. Even caffeine can stimulate it.
And what inhibits it?
Hormones like somatostatin and secretin act as brakes. Also, strong sympathetic stimulation, like during fear or pain, can shut down secretion.
Okay, and pepsin, you said it digests protein.
Yes, secreted as inactive pepsinogen by chief cells, activated by the acid. It starts breaking down proteins into smaller polypeptides, but once the chyme enters the alkaline environment of the duodenum, pepsin gets inactivated again.
Right. So with all this potent acid and enzymes, how does the stomach not digest itself?
The mucosal barrier. This is key. It's a multi-layered defense. Tight junctions between the epithelial cells prevent leakage. There's a thick layer of alkaline mucus coating the surface. And good blood flow helps whisk away any acid that gets through and provides bicarbonate.
But this barrier can be broken.
Unfortunately, yes. Things like infection with the bacterium H. pylori, taking aspirin or NSAIDs regularly, excessive alcohol, or even bile backing up from the duodenum can damage this barrier. That leads to inflammation, gastritis, and potentially painful ulcers.
A delicate balance then. Okay, so we've churned and mixed in the stomach, but the real heavy lifting of absorbing nutrients happens next, right?
Absolutely. That's the main job of our next stop. The incredibly efficient absorption hub, the small intestine.
This is the long one, right?
The longest part of the GI tract, yeah. About 5 to 6 meters coiled up in your abdomen. It's divided into 3 segments. First, the duodenum, which receives the chyme from the stomach.
And secretions from the pancreas and liver.
Exactly. Then comes the jejunum, the middle section, and finally the ileum, which connects to the large intestine via the ileocecal valve. Figure 37.9, A shows these segments laid out.
Yeah.
The jejunum and ileum are kind of suspended by the mesentery, this fan-like membrane holding them in place and carrying their blood vessels and nerves. The whole thing sits within the peritoneal cavity, lined by the peritoneum.
And its structure is all about absorption.
Totally. It's a masterclass in maximizing surface area. First, you have large circular folds in the lining. Then, covering those folds are millions of tiny finger-like projections called villi. Think of them as the functional units.
Okay, villi.
Each villus, as you can see in figure 37.9b, is covered with absorptive cells called enterocytes, along with mucus-producing goblet cells. And then, the surface of each enterocyte is covered in even tinier projections called microvilli.
Wow, folds on folds on folds.
Exactly. The microvilli form what's called the brush border, and it increases the surface area exponentially. It's estimated the total absorptive area is something like 250 square meters.
Like a tennis court inside you.
Pretty much. And at the base of these villi are pits called the crypts of Lieberkühn. These are like stem cell factories. Undifferentiated cells are born here, then they migrate up the villus, maturing as they go, replacing the older cells at the top. The entire epithelial lining turns over every 4 to 7 days.
That's incredibly fast turnover.
It is, which is great for repair, but it also makes the small intestine very sensitive to things that interfere with cell division, like malnutrition or chemotherapy drugs. Nutrient intake actually stimulates this rapid turnover.
So what does all this structure mean for actually digesting the food?
Well, the chyme coming from the stomach is still pretty acidic and only partially digested. In the duodenum, it gets mixed with alkaline pancreatic juice, bile from the liver and gallbladder, and enzymes secreted by the intestinal wall itself. Figure 37.9 gives a nice overview.
What happens then?
Pancreatic enzymes, intestinal brush border enzymes, and bile salts work together to finish the job. Complex carbohydrates are broken down into simple sugars like glucose, proteins are broken down into amino acids, and fats. Well, fats are a bit more complicated.
How so? Let's talk about dietary fat. Maybe box 37.1 helps here.
Yeah, box 37.1 distinguishes different types of fats, saturated, monounsaturated, polyunsaturated, and notes their links to LDL bad and HDL good cholesterol. But the digestion process itself involves four main phases.
Okay, what are they?
First, emulsification and lipolysis. Bile salts break large fat globules into tiny droplets, increasing the surface area for pancreatic lipase to act. Lipase then breaks triglycerides down into fatty acids and monoglycerides. Second, micelle formation. Bile salts surround these fatty acids and monoglycerides, forming tiny water-soluble spheres called micelles.
So the micelles help them get absorbed.
Exactly. That's phase 3, fat absorption. The micelles ferry the fats to the brush border where they diffuse into the enterocytes. Bile salts stay behind and get recycled. Fourth, chylomicron formation. Inside the enterocyte, the fats are reassembled into triglycerides and packaged with proteins into large particles called chylomicrons. These then enter the lymphatic system via the central lacteal and the villus, eventually reaching the bloodstream.
Wow, quite a journey for fats. What about everything else? Where are nutrients mostly absorbed?
The small intestine is definitely the main site. As figure 37.11 shows, almost all the sugars, amino acids, fats, vitamins, and most minerals are absorbed there. About 85–90% of the water you ingest and secrete also gets absorbed in the small intestine, mostly by osmosis following the solutes. Box 37.2 summarizes this well: electrolytes like sodium and potassium, carbs via specific transporters after breakdown by amylases and brush border enzymes, proteins after breakdown by pancreatic proteases like trypsin and brush border peptidases, and minerals like calcium, iron, magnesium, plus of course vitamin B12, eating that intrinsic factor from the stomach.
It really is the absorption hub. And how does it move the chyme along while all this is happening? Intestinal motility.
Two main movements here. First is segmentation. These are localized rhythmic contractions that slosh the chyme back and forth. It's more about mixing than propelling. It ensures the chyme constantly contacts the absorptive surface and mixes well with enzymes.
Okay, mixing. Then how does it move forward?
That's peristalsis. Just like in the esophagus, these are waves of contraction that push the chyme slowly down the intestine. The villi themselves also sway back and forth, helping to stir the fluid near the surface.
Are there reflexes controlling this too?
Oh yes, there are several important ones. The ileogastric reflex. If the ileum gets too full, it sends signals back to slow down stomach emptying.
Makes sense. Prevents overload.
Right. The intestinal reflex. If one part of the intestine gets way overdistended, it inhibits motility in other parts, kind of a protective brake. And the gastrololly reflex. When the stomach is active after a meal, it stimulates motility in the ileum and relaxes the ileocecal valve, pushing contents towards the large intestine. This is probably mediated by hormones like gastrin and cholecystokinin.
And you mentioned something about fasting.
Yeah, during fasting periods, there's a pattern called the interdigestive myoelectric complex, or migrating motor complex. It's like a slow, powerful wave that sweeps down the entire small intestine every 90 minutes or so, clearing out any remaining debris, like intestinal housekeeping.
Keeps things tidy, and then the ileocecal valve opens to let things pass into
the large intestine, our finishing line.
Okay, how long is this part?
Much shorter than the small intestine, about 1.5 meters long. Its main parts, as shown in figure 37.1a, are the cecum, which is a pouch where the ileum joins. The appendix dangles off the cecum, traditionally thought useless, but maybe has immune functions related to gut flora.
Interesting. Then the colon.
Right, the colon, which makes up most of the length, it loops up, ascending colon, across, transverse colon, down, descending colon, makes an S shape, sigmoid colon, and then connects to the rectum, the final straight section ending at the anal canal. The ileocecal valve guards the entrance, and there's another sphincter between the sigmoid colon and rectum.
How is its structure different from the small intestine?
Quite different. No villi here. Instead, the outer longitudinal muscle layer isn't continuous. It's gathered into three distinct bands called tenaeoli.
Tenaeoli.
Yes. And because these bands are shorter than the colon tube itself, they cause the wall to bulge out into pouches called haustra, so it looks segmented. Inside, there are crypts, similar to the small intestine, but mostly packed with mucus-secreting goblet cells and columnar epithelial cells specialized for water absorption. Figure 37.2b shows a cross-section, highlighting the lack of villi, and the presence of tenaeoli looks quite distinct.
Got it. And motility here, is it just pushing waste out?
It's a bit slower. There are segmental movements, primarily in the cecum and ascending colon. These are slow churning motions within the haustra that mix the contents and expose them to the mucosa for water and electrolyte absorption. Think of it as gently massaging the fecal mass.
And peristalsis.
Yes, there are also peristaltic movements that propel the contents along, but they're less frequent than in the small intestine. The key movement here is mass propulsion, usually triggered by the gastrocolic reflex.
Ah, the one that makes you need the bathroom after eating.
That's the one. When food enters the stomach, or chyme enters the duodenum, it triggers strong waves of contraction starting in the transverse colon, pushing fecal matter rapidly into the descending and sigmoid colon and into the rectum, initiating the urge to defecate.
So the main job here is water absorption.
Primarily, yes. Absorbing the remaining water and electrolytes, like sodium and chloride, is the large intestine's main contribution to digestion. Aldosterone can act here to increase sodium and water absorption. It doesn't really absorb nutrients like sugars or amino acids, though it can absorb some short-chain fatty acids produced by bacteria. What's left is consolidated into feces.
Okay, feces formation, then the final step, defecation.
Right. This is initiated by the defecation reflex, also called the rectus-enteric reflex. When feces distend the rectal wall, stretch receptors send signals to the spinal cord.
And what happens then?
This triggers relaxation of the internal anal sphincter, which is involuntary smooth muscle. At the same time, you perceive the urge to defecate.
But we can control it.
Yes, thankfully. The external anal sphincter is skeletal muscle and under voluntary control. If the time isn't right, you can consciously keep it contracted. If you decide to go, you relax the external sphincter and often employ the Valsalva maneuver, taking a deep breath, closing the glottis, and contracting abdominal muscles to increase intra-abdominal pressure, helping to expel the feces.
Right. Okay, so that covers the main path. But you mentioned immunity earlier. The GI tract is a major immune organ.
Hugely important. What's truly fascinating here is the gut-associated lymphoid tissue, or GALT. It's one of the largest lymphoid organs in the body.
GLLT, what does it include?
Key components include specialized cells like Canis cells, found in the crypts of the small intestine. They secrete antimicrobial peptides like defensins and enzymes like lysozyme, helping to control the microbial population.
Okay, Paneth cells. And
then there are pyre patches. These are large clusters or nodules of lymphoid tissue packed with lymphocytes, plasma cells, and macrophages. They're especially prominent in the ileum.
what do pear patches do?
They are critical immune surveillance centers. They sample antigens from the gut lumen, process them, and mount immune responses against pathogens. But they also play a huge role in developing oral tolerance, teaching your immune system not to react aggressively to harmless food antigens or beneficial bacteria.
So defense and tolerance, very important.
Absolutely.
And speaking of bacteria, beyond our own cells, we have this whole ecosystem living inside us, right? The intestinal microbiome.
Yes. Our tiny allies. It's a vast and incredibly diverse community of bacteria, fungi, and viruses living primarily in our colon. The sheer number of microbial cells actually rivals the number of human cells in our body.
Wow. What influences who lives there?
Many things. Our genetics play a role, but diet is huge. Medications, especially antibiotics, can drastically alter it. Age also affects it.
And what do these microbes do for us?
So much. They play critical roles in metabolizing things we can't digest ourselves, like certain fibers, producing short-chain fatty acids, which nourish colon cells. They help metabolize bile salts, hormones, lipids. They synthesize essential vitamins, like vitamin K and some B vitamins.
Okay, metabolic help. What else?
They produce antimicrobial peptides to help keep pathogens out. They help break down potential toxins. They train and activate our immune system, contributing to that GALT function we just discussed. They basically form a protective barrier against harmful invaders.
So they're really integrated into our health. How does this microbiome develop?
We're born essentially sterile. Colonization starts rapidly within hours or days of birth, influenced by delivery method, feeding, environment. It becomes more complex over the first few years, stabilizes in adulthood, and then tends to decline somewhat in diversity with aging. The distribution varies too. The stomach's acid keeps numbers low. The duodenum and jejunum suppress growth. But the colon is teeming with mostly anaerobic bacteria.
Incredible.
Right.
Okay, running all this machinery, the tract, the immune cells, the microbes requires resources. That brings us to splanchnic blood flow.
Right, the supply line. This term refers to the blood flow to all the abdominal digestive organs, stomach, intestine, spleen, pancreas, liver. It's a significant portion of your total cardiac output, especially after a meal.
How is it controlled?
It's regulated by a combination of factors. Overall cardiac output, the autonomic nervous system, sympathetic nerves generally constrict vessels, parasympathetic nerves indirectly increase flow via increased activity, various hormones released during digestion, and local autoregulation based on metabolic needs.
And it serves another purpose, too.
Yes, crucially, the splanchnic circulation acts as a major blood reservoir. If your body needs to increase blood pressure quickly, like during exercise or shock, sympathetic nerves can constrict these vessels, shunting a large volume of blood back into the general circulation for your heart, brain, and muscles. That figure 37.6 we mentioned earlier really illustrates how extensive this network is.
Very clever design. Now, beyond the main tube, we have the essential helpers, the accessory organs of digestion.
The liver, gallbladder, and the exocrine pancreas. They aren't part of the GI tract tube itself, but they produce and secrete substances absolutely essential for digestion.
And they all deliver their goods to the same place.
Pretty much. Their secretions, bile from the liver and gallbladder, enzymes and bicarbonate from the pancreas, typically enter the duodenum through a common gateway controlled by a muscle called the sphincter of Oddi. Figure 37.32 shows their location and how they connect near the start of the small intestine.
And the liver. You said it's a multitasker.
Oh, absolutely. Its role goes way beyond just making bile for digestion. It's central to metabolism, blood clotting, storing blood, and immunity. It really is the body's central processing unit.
Okay, let's dive deeper into the liver. It's a big organ, right?
The largest solid organ, yeah. Weighs about 1.2 to 1.6 kilograms. It sits mainly in the upper right part of your abdomen, just under the diaphragm. It has right and left lobes, held in place by ligaments like the falciform ligament, and it's covered by a connective tissue layer called Glisson's capsule.
Is that capsule important?
It contains blood vessels, lymphatics, and nerves. It's also sensitive to stretching or inflammation, which is why liver enlargement or hepatitis can cause that dull, aching pain in the upper right abdomen. Figure 37.14 shows the lobes and ligaments clearly.
You mentioned its blood supply is unique.
Very unique. It gets a dual supply. The hepatic artery, branching off the aorta, brings oxygenated blood, supplying about 25% of the liver's flow. But the real workhorse is the hepatic portal vein.
Portal vein. That's the one coming from the intestines.
Exactly. It carries nutrient-rich but deoxygenated blood collected from the entire stomach, intestines, spleen, and pancreas directly to the liver. This accounts for about 70% of the liver's blood flow. Figure 37.15 illustrates this portal system beautifully. It means virtually everything absorbed from your gut passes through the liver first for processing before entering the general circulation.
A critical checkpoint. What about the liver's internal structure? Lobules.
Right. The functional units are called liver lobules. You can picture them, as in figure 37.16, as roughly hexagonal structures. At the center of each lobule is a central vein. Radiating out from that are plates or cords of the main liver cells, the hepatocytes.
Hepatocytes, those are the workhorse cells.
They are. They perform most of the liver's metabolic functions, bile production, detoxification, and they can regenerate, which is amazing. Between these cords of hepatocytes are specialized capillaries called sinusoids.
Sinusoids, different from regular capillaries.
Yes, they're wider and leakier. This is where the oxygenated blood from the hepatic artery mixes with the nutrient-rich blood for the portal vein, allowing the hepatocytes intimate contact with everything coming in. Lining these sinusoids are other important cells. Endothelial cells, of course, but also Kupffer cells, which are resident macrophages, big eaters, part of the immune system, cleaning up debris, bacteria, old red blood cells. They're key in bilirubin production and injury response. They're also stellate cells, which normally store vitamin A but can become activated in injury and contribute to fibrosis or scarring, and natural killer cells for tumor defense. It's a complex microenvironment.
Okay, here's what gets really interesting. Bile secretion. The liver makes a lot of this stuff.
It does, around 700 to 1,200 milliliters of this alkaline, yellowish-green fluid called bile every single day.
What's in bile?
Its key components for digestion are bile salts, which are derived from cholesterol. It also contains cholesterol itself, bilirubin, that breakdown product of red blood cells, electrolytes, and water.
And where does it go?
Hepatocytes produce bile and secrete it into tiny channels between them called bile canaliculi. These merge into larger bile ducts within the liver, eventually forming the common hepatic duct. This usually joins the cystic duct from the gallbladder to form the common bile duct, which carries bile down to the duodenum, entering through that sphincter of Oddi.
And the main job of bile salts is?
Fat emulsification and absorption. They act like detergents, breaking down large fat globules into smaller ones, increasing the surface area for enzymes like lipase to work. They also help form those micelles we talked about, essential for absorbing fatty acids and fat-soluble vitamins.
You mentioned recycling bile salts.
Yes, the enterohepatic circulation. This is super efficient. After bile salts do their job in the duodenum and jejunum, about 95% of them are actively reabsorbed in the terminal ileum. They travel back to the liver via the portal vein, are taken up by hepatocytes, and get secreted into bile again. Figure 37.17 shows this recycling loop. Each bile salt molecule might cycle through 10-20 times during the digestion of a single fatty meal.
Very economical. Are there different kinds of bile acids?
Yes, the liver synthesizes primary bile acids, cholic and chenodeoxycholic acid. Gut bacteria then modify some of these into secondary bile acids. Before secretion, the liver conjugates most bile acids with amino acids, glycine or taurine, which makes them water-soluble and better detergents. The process of bile secretion is called choleresis, and things that stimulate it like bile salts themselves, CCK, vagal stimulation, and secretin are called choleretics.
Okay. And bilirubin metabolism, that's linked to red blood cells.
Right. It's a byproduct of breaking down the heme portion of hemoglobin from aged red blood cells, mostly done by macrophages like Kupffer cells. Heme gets converted first to biliverdin, green, then to unconjugated bilirubin, yellow. This form is lipid-soluble and not easily excreted, so it binds to albumin in the blood and travels to the liver.
What happens in the liver?
Hepatocytes take up the unconjugated bilirubin and conjugate it, attach glucuronic acid, making a water-soluble conjugated bilirubin. This form can be excreted into the bile. Figure 37.18 details these steps.
And then it leaves in bile.
Yes. Once in the intestine, gut bacteria converted conjugated bilirubin into urobilinogen. Most urobilinogen is further converted to stercobilin, which gives feces their characteristic brown color. A small amount of urobilinogen is reabsorbed, enters the bloodstream, and is excreted by the kidneys as urobilin, contributing to urine's yellow color.
So bilirubin levels tell us about liver function and red blood cell breakdown.
Exactly. Problems with any step in this pathway can lead to jaundice, that yellowing of the skin and eyes.
Beyond bile and bilirubin, what are other key liver functions, vascular and hematologic?
Yes, the liver stores a significant amount of blood, helping regulate blood volume. It synthesizes almost all the essential plasma proteins, including albumin and, crucially, the clotting factors. Vitamin K is needed for the synthesis of several clotting factors, and the liver plays a role there too.
And metabolism.
Huge. It's central to carbohydrate metabolism, storing glucose as glycogen, making new glucose, protein metabolism, synthesizing non-essential amino acids, converting ammonia to urea, and fat metabolism, synthesizing lipoproteins, cholesterol, phospholipids.
And detoxification.
Metabolic detoxification, or biotransformation, is critical. The liver modifies chemicals, both foreign ones like drugs and toxins, and internal ones like hormones, usually making them less toxic and easier to excrete, often via bile or urine. This involves complex enzyme systems like cytochrome P450.
Is that where drug interactions happen?
Often, yes. And it's why the liver is susceptible to damage from certain drugs or toxins. The Did You Know box gives the example of paracetamol, or acetaminophen. Normally, the liver safely detoxifies it. But in an overdose, or sometimes with chronic use, especially combined with alcohol, the normal pathways get overwhelmed. A toxic metabolite builds up and can cause severe liver damage, even acute liver failure. It really highlights the liver's vital and vulnerable detox role. It also stores vitamins like A, D, E, K, B12, and minerals like iron and copper.
A true powerhouse. So the liver makes bile. Where does it go between meals?
Into the gallbladder. This is a small, pear-shaped, sac-like organ tucked neatly underneath the liver. You can see it in figure 37.19.
That's its job, just storage.
Its primary function is to store and concentrate bile between meals. When the sphincter of Oddi is closed, which it is during fasting, bile produced by the liver backs up through the cystic duct into the gallbladder.
And it concentrates it.
Yes, the gallbladder wall actively absorbs water and electrolytes from the bile, making it much more concentrated, sometimes 5 to 10 times more concentrated than liver bile.
Then what triggers its release?
After you eat, especially a meal containing fats or proteins, chyme entering the duodenum triggers the release of the hormone cholecystokinin, CCK. CCK is the main signal for the gallbladder to contract strongly. Vagal nerve stimulation also plays a role. This contraction forces the stored concentrated bile out through the cystic duct, down the common bile duct, through the now relaxed sphincter of Oddi, and into the duodenum to help digest the meal.
Got it. Bile's reservoir and concentrator. Now, the last accessory organ, the exocrine pancreas.
Right. The pancreas is interesting because it has both endocrine functions, making hormones like insulin and glucagon released into the blood, and exocrine functions, making digestive juices released into ducts. This chapter focuses on the exocrine part.
Okay, what does the exocrine pancreas look like?
It's composed of clusters of secretory cells called acini. These acinar cells produce the potent digestive enzymes. Leading away from the acini are ducts, lined by cells that secrete large volumes of alkaline bicarbonate-rich fluid. These small ducts merge into larger ones, eventually forming the main pancreatic duct, which usually joins the common bile duct just before entering the duodenum at the sphincter of Oddi. Figure 37.19 shows these acinar cells and the duct system.
And that alkaline fluid is important.
Absolutely crucial. The pancreatic juice is highly alkaline, with a pH around 8. This is needed to neutralize the acidic chyme arriving from the stomach. Remember, stomach enzymes like pepsin stop working in alkaline conditions, but pancreatic enzymes need this neutral or slightly alkaline environment to function optimally.
What are the key pancreatic enzymes?
There are enzymes for digesting all major food types. Proteases like trypsinogen and chymotrypsinogen break down proteins, pancreatic amylase breaks down carbohydrates, and pancreatic lipase breaks down fats, triglycerides. There are others too, like elastase and carboxypeptidase.
You said trypsinogen. Ogen means inactive.
Exactly. This is a critical safety feature. The powerful proteases are secreted as inactive proenzymes or zymogens, like trypsinogen, chymotrypsinogen. If they were active inside the pancreas, they'd start digesting the pancreas itself, leading to pancreatitis, which is very dangerous.
So how do they get activated?
Activation happens only once they reach the duodenum. An enzyme embedded in the duodenal brush border, called enterokinase or enteropeptidase, specifically activates trypsinogen into active trypsin. Trypsin then acts as a master switch, activating all the other pancreatic proenzymes, including more trypsinogen. It's a controlled cascade reaction, safely confined to the gut lumen.
Clever system. How is pancreatic secretion regulated?
It's mainly controlled by hormones released from the duodenum in response to chyme. Secretin is released when acidic chyme arrives. Its main job is to stimulate the pancreatic duct cells to secrete lots of that bicarbonate-rich fluid to neutralize the acid. Cholecystokinin, CCK, is released mainly in response to fats and proteins. Its primary role is to stimulate the acinar cells to release large quantities of digestive enzymes. Acetylcholine from vagal nerve endings also stimulates enzyme secretion.
So acid triggers bicarbonate, fats, proteins trigger enzymes. Makes perfect sense.
It's a beautifully coordinated system.
It really is. Now, we know this system is incredibly robust, but like any finely tuned machine, it must experience changes over time, especially with aging. What are some common ways digestion shifts as we get older?
That's a crucial point, and the geriatric considerations section highlights several key areas. Things often start right in the mouth. Tooth loss or deterioration is common, making chewing difficult. There's often a decline in the senses of taste and smell, which can decrease appetite and enjoyment of food.
And saliva.
Saliva production can decrease, leading to dry mouth, xerostomia, which makes swallowing harder, dysphagia, and increases risk of cavities. Moving down, esophageal motility might slow slightly.
What about the stomach?
Stomach motility and emptying can slow down. Blood flow might decrease. There's often reduced secretion of gastric juice, particularly acid, aquahydria, or hypochlorhydria, which can affect B12 and iron absorption and increase bacterial overgrowth risk. The protective mucosal barrier may also decline in integrity.
And the intestines.
Changes in the gut microbiota composition are common. Mucosal immunity, particularly the function of pear patches, may decline. There can be disruptions in the brain-gut axis communication. Structurally, villi might become shorter or broader, potentially reducing absorptive surface area slightly. Intestinal motility generally slows, contributing to the increased prevalence of constipation in older adults. Fecal incontinence can also become more common due to weakened sphincter muscles or nerve dysfunction.
Liver and pancreas.
The liver often decreases slightly in size and blood flow. Its regenerative capacity might slow, and its ability to metabolize some drugs can decrease, which is important for medication dosing. The pancreas may undergo some fibrosis and atrophy, potentially leading to reduced secretion of digestive enzymes, although usually not enough to cause significant malabsorption unless there's underlying disease. Gallstones also become much more common with age.
So a general trend towards things slowing down or becoming less efficient?
Generally, yes, but it's highly variable between individuals. Lifestyle factors, diet, medications, and overall health play huge roles. But these age-related changes definitely contribute to some of the common digestive complaints and nutritional challenges seen in older populations.
Okay, so what does this all mean for our understanding? We've taken quite a journey, from the first bite, through the churning stomach, the amazing absorption in the small intestine, final processing in the large intestine.
Explored the crucial helpers, liver, gallbladder, pancreas.
Uncovered the powerful immune functions, met our tiny microbial allies, It's truly an amazing coordinated symphony happening inside us constantly, just to process food, get nutrients, and keep us healthy.
It really is. And if we connect this to the bigger picture, well, understanding this delicate balance, these intricate feedback loops within the digestive system, it really helps us appreciate the fragility of health, doesn't it?
Yeah.
It underscores how profoundly our lifestyle choices, our diet, stress levels, medications impact these systems. And it highlights the potential for things to go wrong, for pathology to develop. When any of these finely tuned processes get disrupted, it makes you realize digestion isn't just, a mechanical process. It's absolutely fundamental to our overall well-being.
That's a great final thought. Digestion as a fundamental pillar of health. Absolutely. Well, thank you for joining us on the deep dive into the fascinating, complex world of the digestive system.
My pleasure.
We really hope this journey sparked some curiosity. Keep exploring the wonders within and around us.